The Venus Flytrap is a marvel of precision. Its trap is a living mechanism built for accuracy, not randomness. Each leaf is split into two lobes that stay open, waiting for prey. The plant waits until something touches its inner trigger hairs more than once. That second contact changes everything, triggering the trap to snap shut almost instantly. The trap seals the insect inside with interlocking edges. At first, there are small gaps, allowing tiny insects to escape, but if the prey keeps moving, the trap tightens further. Once sealed, the plant begins digestion. Enzymes slowly break the insect down, turning it into nutrients the plant can actually use. This system is fast, but more importantly, it is deliberate. The plant avoids reacting to anything that doesn't matter, saving energy for real opportunities. Each trap can only close a limited number of times, so every decision has weight. Over time, used traps darken and die, while new ones grow from the center, ready to repeat the same process. The whole plant runs on timing and efficiency, not constant action. It never chases, never moves across the ground, yet it still captures living prey. In the end, it feels less like something passive and more like something that simply waits until it's time. Next, we have the Nepenthes, or pitcher plant. Its trap doesn't snap, it invites. The leaf extends into a deep, hanging pitcher filled with liquid. With a rim that looks almost harmless at first glance, that rim is the key. It becomes extremely slippery, especially when wet, making it nearly impossible for insects to keep their footing. Attracted by nectar, they move closer, lose grip, and fall straight inside. The inner walls are smooth and angled downward, so climbing back out is not an option. What looks like a container is actually a one-way drop. Inside, the liquid is not just water, it's a digestive mix that slowly breaks down anything that falls in. From small insects to, in some cases, much larger prey, the plant doesn't rush the process, it lets time do the work. Instead of reacting quickly, it relies on design. Everything about the structure guides the prey toward a single outcome. It doesn't need speed because once something slips, there's no way back. Now, let's look at the sundew. Its trap looks almost delicate, covered in tiny hairs tipped with glistening droplets that resemble morning dew. But those droplets are not water. They are thick, sticky, and impossible for small insects to escape once touched. The moment an insect lands, it becomes stuck. Struggling only makes it worse. The surrounding tentacles begin to bend inward, slowly wrapping around the prey. What starts as a light contact turns into full enclosure. The movement is slow, but it's constant and intentional. Over time, more tentacles join in, securing the prey and increasing contact. Digestive enzymes are released. After the process is complete, the tentacles gradually return to their original position, ready to trap again. It doesn't rely on speed or force, but on patience and control. The shine that attracts prey is the same thing that seals its fate. What looks harmless at a distance becomes inescapable up close. Finally, we have the bladderwort. Its trap is completely hidden, working underwater or within wet soil, where nothing is visible from the outside. Instead of leaves that snap or surfaces that trap, it uses tiny bladder-like sacs that act as suction devices. Each bladder stays under tension, sealed shut and ready. Along the entrance, there are fine trigger hairs that detect movement. When something small brushes against them, the door opens inward in a fraction of a second. Inside the bladder, there is no escape. The prey is held in a sealed chamber where digestion begins almost immediately. The plant absorbs nutrients directly from what it captures. After each capture, the bladder resets itself, pumping water out to rebuild the pressure needed for the next trigger. This cycle repeats continuously, trap after trap. Among carnivorous plants, this is one of the most mechanical and efficient systems. It doesn't lure or chase, it simply waits for the exact moment something gets too close. Now, let's look at the cobra lily. Its trap doesn't look aggressive at first, but the shape tells a different story. The leaf curves upward into a hollow structure. Light filters through translucent patches at the top, creating bright spots inside the trap. For an insect, it feels like a way out, but it isn't. As it moves toward the light, it hits solid walls again and again, losing direction. The real entrance is below, and once the insect slips inside, the path becomes confusing. The inner surface is smooth and lined with downward-pointing structures that make climbing back almost impossible. What seems open and bright is actually a maze designed to mislead. There is no quick snap or sudden closure here, only gradual exhaustion. The insect keeps moving, searching for an exit that doesn't exist, until it eventually falls deeper. At the bottom, digestive processes begin, supported not just by the plant, but also by microorganisms living inside the fluid. Together, they break down the prey over time. The plant relies on deception rather than speed, guiding movement instead of forcing it. Each part of the structure plays a role in that illusion. And by the time the insect realizes something is wrong, it's already too far inside to escape. Finally, we have the bladderwort. Its trap doesn't move suddenly or look threatening. It just sits there with a smooth, glossy surface that seems harmless. The leaves are covered in a thin layer of sticky fluid that glistens under the light, attracting small insects that land without hesitation. The moment they touch it, they're stuck. The surface holds them in place, and even the smallest movement only spreads the contact further. There's no snapping or closing, just a quiet capture that happens instantly. What looks like a simple leaf is actually a fully active trapping surface. As the insect struggles, the leaf responds slowly. The edges can curl inward slightly. Digestive enzymes are released across the surface, breaking the prey down right where it lies. The plant absorbs nutrients directly through the leaf. After digestion, what remains is minimal, often just a dry outline left behind. The leaf stays in place, ready to trap again, maintaining the same passive appearance. It relies entirely on contact and adhesion, turning stillness into an effective strategy. There is no urgency in how it works, only consistency. Now, let's look at the cephalotus. Its trap looks compact, almost controlled, but everything about it is built to pull prey inward. The pitchers sit low to the ground, with a wide opening and a thick ridged rim that secretes nectar. That rim becomes slippery, especially in humid conditions. Once they slip, they drop into a deep chamber lined with smooth walls that offer no grip. The lid above doesn't close, but it helps control moisture. Inside, the environment changes. The lower section holds digestive fluid that slowly breaks down whatever falls in. Fine internal structures prevent any upward movement. The plant doesn't rely on speed or movement, it relies on positioning and structure. Each pitcher functions independently, capturing and digesting over time. As older traps wear out, new ones grow alongside them, maintaining the cycle. It stays still, but every part of it is active in its own way. Finally, we have the genlisea. Its trap works entirely out of sight, buried in wet soil where nothing looks unusual from above. Instead of visible leaves doing the capture, it uses thin, tubular structures that extend underground. These tubes split into spiraling arms, forming a narrow path. At the entrance, everything seems open, but the deeper it goes, the tighter and more restrictive it becomes. Tiny inward-pointing hairs line the interior. Small organisms enter easily, but once inside, turning back is no longer possible. As the prey moves deeper, it's gradually forced toward the central chamber. There, digestion begins, breaking down microorganisms that the plant absorbs as nutrients. The entire system functions without any visible motion. Each trap is passive on the outside, but internally it's highly controlled. It's one of the few carnivorous plants that hunts where you can't see it at all. Thanks for watching. If you made it this far, you already know what to do.